welcome Dr. Rebecca Forkner, who is an entomologist at George Mason University. And she's also a community ecologist and plant biochemist. She researches the ecological and evolutionary factors that are responsible for the colors that we observe in plants and insects, and she researches plant-insect interactions. So I know we're all going to learn a lot tonight, and I look forward to this talk. So I have no further comments really, and I'm ready to turn it over to Dr. Fortner.
Okay, great. Thanks, Glenda. I can't tell you, I'm so excited that Glenda asked me to talk about caterpillars because the truth is when I give seminars, I almost never get to talk about the organisms themselves. In fact, most of my talks are very dry graphs and slides and research outcomes. So people often kind of really ignore what's going on behind the scenes with the organisms themselves. And I also rarely get asked about the caterpillars as opposed to the adult form, the butterflies, which a lot of the research that my students do is focused on their colors and morphology and wing scales. So what I'm going to do today is just focus on the juvenile, the larval instars of butterflies and moths. What I want to do is dispel some of the myths that I think are maybe not myths, but I would say misconceptions, I guess, that people have about this particular group. And I'm going to show you about 30 years worth of slides at this point of me photographing my cuticles as I try to get close-up photos on most of these organisms. So I guess one of the things that you'll notice is that a lot of these photographs are close-ups, macro photography, my thumbnails aside here. And that's because most of what we're talking about out of these maybe 174,000 species are super tiny things. So when we kind of think about the sort of if I'm teaching an undergraduate class or I'm taking students out in the field for the first time, this is what they are expecting to see. So they are often disappointed if we don't automatically find a giant Cecropia moth caterpillar that's chowing down on a plant leaf. And I do think that we have this bias in the popular press to really portray things that are charismatic. So we do that for vertebrates, we do it for insects as well. So what I want to do is really start here with what I refer to as the Eric Carlisle effect, the Very Hungry Caterpillar book, which we all read as a child. So it's probably the most overused reference in my student talks, and they're all really focused here on this idea that the things that we see for the juvenile stages of butterflies and moths are voracious plant feeders, and we tend to think of them almost exclusively as things that are plant feeders, so and things that are large. And I don't want to underscore or sort of undermine your sort of fascination with the fact that they can get really large. This is Citharona regalis, the hickory horn devil, which is the largest one in the United States, and it does get pretty big. This change that it undergoes from the egg to the final instar, the penultimate instar, before it undergoes that metamorphosis is extreme. So if we think about this in terms of maybe if we had to convert this to how big I might have to be by comparison to make this kind of change from this first instar that we see all the way on the left here to the largest one on the right. That's about a change from maybe 3.5 milligrams to about 10 grams. So if we think of that in human terms, that would be like me. I weighed slightly more than 4 pounds premature, slightly more than 4 pounds at birth. That would be like me weighing about 5 metric tons now, just getting to sort of the middle preteen stage before I became an adult. So it's an enormous change that they do undergo. But this idea that they're all really large, like this hickory horn devil, is probably a sort of misconception. And I think we also have this idea generally when we're talking about caterpillars that we're thinking that as they form, undergo the metamorphosis, form the chrysalis, and become an adult, they're all going to be something really beautiful and showy that is nectar feeding as well. So we have this idea that they're plant feeders and chewing through these plants at a fast rate and getting really big as larvae, and then there are these beautiful butterflies, this sort of Disney Pixar effect here. Not to sort of talk down on these movies. I think some of the entomologists that are sort of in the background here are doing some great jobs and leaving lots of little entomology jokes in there. But I really want to convey that what you would see, even if you do go out now, would be what my colleague John Lill refers to as LBJs, little brown jobs, because most of the adults are going to be super tiny moths, and most of the caterpillars are going to be something that's tiny and green. So this slide really shows more of what we would see if we were hunting caterpillars right now in Northern Virginia forests. So to understand really how we get here from this idea that everything is really a monarch and some beautiful butterfly, we really want to start with what is the kind of the most common thing that you might see. And it really isn't something large. It's something like this chestnut leaf miner. So this scale bar in the bottom here for the moth is only a millimeter. So our hickory horned would be the largest, and this chestnut miner would be one of the smallest things that we might find. And it's more closer to the actual average size of a moth that we would see. And they don't all necessarily leave these nice little trails as they feed internally on the leaf either. Most of them are pretty cryptic and hard to find. So to understand really how we get, maybe 92% of our butterflies and moths being something fairly tiny and obscure, we need to go back a little bit to some of our fossil organisms and try to take a look at the evolutionary history. So what you're looking at is this sort of radial circular tree of life. Um, I'll note that it says on the left here that about 85% of our Lepidoptera, our butterflies and moths, are the moth form. And in particular, it's both. We're going to sort of break this into two. So looking at the upper part of this tree, we have the sort of primitive moths. The butterflies are there in yellow. You'll notice it says diurnal butterflies there at the bottom in the legend. And then the remainder of this are the more recently evolved moths. So there's a few things about this that I want you to recall. It won't be a quiz at the end like I do with my students. The first is that most of what we are going to see is going to be a tiny moth, not a large butterfly. So that's about 85%. But also, if we look at the timing of this, we really if we know anything about entomology, we might have in the back of our minds that butterflies radiated with flowering plants. We think of them, like I said, we think of them as nectar feeders. But our fossil record actually for insect goes back further than the angiosperms. So we think that they originated about 300 million years ago. But this association that we have with nectar feeding, the proboscis took a long time to evolve, about somewhere between 150 million, 175 million years after the origin of butterflies, sort of around 300 million years ago. So that is a more derived, more recent form. And really what we see in this upper portion of this graph is actually things that don't have that coiled proboscis in the adult. They have the chewing mouth parts of the larvae. The other thing I want you to notice here is that there is a big radiation that does occur along with the angiosperms after the butterflies. That's going to be the vast majority of our species. There's at least, for example, in this last family here, the Noctuoideae, at least 70,000 species. By comparison, our butterflies are somewhere around 16,000 species in all. Now, there's a few other things in this graph I won't have time to go into today, and one of those is the little red dots that are on here are the origin in the adult of hearing organs that were initially thought to be for sort of counter echolocation, sound producing or hearing organs to avoid being consumed by bats. The bats are there in that blue inner circle. That's when we think those evolved. So really what we're seeing is that some of those sound producing and hearing organs were actually present earlier than bats. And we're going to find that they're used for a variety of other things. And I hope I'll get a chance to talk about some of that today. So our butterflies have originated from these night flying tiny moths. And I want to show you a little bit more of what those are going to look like. So the first thing you should know from that sort of phylogeny, that tree of life, is that the chewing mouth parts are actually ancestral. And we don't get these really advanced coiled proboscis until much later. So looking at the caterpillar, this is an up-close picture of what the caterpillar face looks like in macro scale. They do have antennae. They're sort of surrounding the mandibles on the outside. They're much smaller than the elongated antenna in the adults. They have what are called stemata. I'm going to talk about those a little bit later. We thought these were fairly simple seeing organs that mostly just let the caterpillar see where the horizon was in case it fell out of a tree. Turns out they have many more sensory organs than that for sensing light, so I'll talk about those. But I really put this here so you can see what the mandibles look like. Because our first group of moths that we're going to talk about are ones that are chewing, and they're actually feeding in these very primitive groups on soil, fungi, dead tissue, and waste. And so there's quite a number of species, at least 800 in the micro moths in this first bar here, that are feeding in the soil. So if you think about them feeding first on non-vascular plants that would have been around prior to flowering plants, it really does make sense. So in this I've sort of changed the tree of life here a little bit to sort of be more in the linear way that we tend to think about these. Each of these numbers 1 through 6 here is a step in the musculature evolution of that proboscis. So you see that the butterflies really don't come along till we're way up here. And we have all of these other forms of moths here with caterpillars that are feeding in different ways. And they're not necessarily all feeding on green leaves. So I want to talk about some of those, and we're going to start with those that are primitive and have those chewing mouth parts. So if you are out and about today, you might actually be able to find some of these. These are what we refer to as the Micropterygii, the moss eaters. So I had a graduate student that I met when I was teaching in Missouri, who was studying bryophytes, and he said, "Oh, there's no herbivory on bryophytes. There's nothing that eats them." And I said, "I think I could find some." So this is this particular group. The caterpillars are shown in the center here. Those are the eggs on the very bottom. The adults of these do have those chewing mouth parts. They're going to be feeding on pollen. But the larvae would be feeding in the soil on fungi, but also potentially on bryophytes, mosses, liverworts, and some of the primitive plants that would have been present. Now, as we go up from this group, we are changing the mouth parts a little bit and starting to evolve for the adults, something that's a little bit more like that coiled proboscis. But really what we have as we sort of move up in sort of advancements and get closer to modern day butterflies and moths are the root feeders. In the top is one of the, I've included this because it holds the record for being the most fecund female ever. This particular ghost moth or Bardi moth, sometimes also referred to as a hepaliid. These are root feeders. So cicadas aren't the only things feeding underground. These actually build small tunnels, feed on some of the softer roots. The Australian ghost moth here, the rain moth is another name for this. This particular female was laid 29,000 eggs in a single day. Once it was dissected, she had another 15,000 she could have laid. And when they're flying about, they just sort of toss their eggs out as they fly. So they land wherever on the ground, and the caterpillars hatch and burrow into the soil. Now we do have some of these here within Virginia, so they're in the Vitaceae. These are root feeders on things like grapes, so they can be pests for viniculture. The adults in this case look a little bit more like a wasp, and that is the Sasiid family as well as the Hepaliid. So we have some root feeders early on as well. Now, I don't want to convey that all these root feeders are pests or negative things. So I want to mention that in terms of medicinal value, the most expensive or considered to be sort of the most return financially return on investment here is one of these Hepaliid ghost moths. This is a Tibetan ghost moth. The caterpillar's in the center there. The Ophiocordyceps is the fungus that grows on this after it's been feeding underground for a while. So they do get infected with fungus. And this is sold, sometimes referred to as Tibetan Viagra. It's sold as an aphrodisiac. It goes for $130,000 per kilogram. Just so you know, by comparison, I checked the price of gold. Gold goes for about 85,000 per kilogram. So this was enough in some cases. If you heard the NPR story, there were some fatal fights over access to some of these caterpillars and their fungi. So these would be in that Hepaliid as well, along with that ghost moth, feeding underground on roots. Then we get to things that are still essentially out there and about. We are getting to the evolution of mammals, but we're not necessarily seeing a lot yet of the sort of plant feeding that we typically associate with butterflies and moths. Instead, what we have are things that are starting to feed on animal tissue. So I put an image up here just to remind you to put some cedar in your closet this time of year to save your wool sweaters. So we would have seen things in the Tennaid, the clothes moss, the carpet moss is another name for this group. The caterpillar is shown there along with an empty pupal case for this particular one. And you can also see in the center of the image some of the caterpillar waste. So they're really good at breaking down hair, chitinous material. My students who are in the forensics program, where we've just started a body farm, are finding this particular family on the bone stage for the corpses that they're watching decay. Now, what you might not also know is that this group actually plays a pretty substantial role in decomposing a lot of animal tissue. So this is one, this ceratophagia here, that makes the caterpillar makes little tunnels on the back of old turtle shells. You can see that there have been several feeding on this particular shell. This is some of Mark's work from 2005 showing this particular species. So we start to see that a lot of things move from the soil feeding or root feeding to feeding on mammal tissue. And then we get to things that are a little bit more along the lines of what we think of for modern day butterflies and moths. The adults have the coiled proboscis here. These are twirler moths and the Gallicaid, but these would still be the sort of bryophyte and moss feeding. You can see in the upper image some of the silk casing this caterpillar has made just to crawl over the top of these bryophytes. And again, in the bottom, what you're looking at is a little shelter that it's made from its own waste and silk. These are going to be about the same size as those clothes moths that are feeding in your, hopefully not feeding in your closet. And then we move into ones that are scraping algae potentially off of some of the sloths. So this is a colleague, Chris Miller, and he's looking at a three-toed sloth here and picking some of the caterpillars off of its back. There are some that feed on the algae that grow on the fur. If you are familiar with sloths, they're resting in the tree canopy. They have these epiphyls that are growing on their fur to make them even more camouflaged. And we're starting to see within this group things that are feeding not just on the algae and on the hair, but also on the waste of the organism. So in the upper right, what you have is an anthill parrot moth. These actually will sit in the nest of the parrot. The parrot nests in termite mounds, and it's feeding within the waste and the hair, the feathers, and cleaning and trying to keep the nest clean. So it's considered to be sort of a commensal relationship. And you can see the caterpillar there in the center, right next to a penny, showing you the size. The moths are really funny. They have this sort of posture of sort of standing up really straight. The adults would finally be in this particular group things that are nectar feeding. Now, one of the things that I want to do before we get into talking about more of the modern day caterpillars is mention that what we would also have seen evolving is a predatory lifestyle. So we have the Eupathisia, which are a group of geometrids of inchworms that are predatory. We have the Hypocosmia, which is a snail feeder, so it will actually use silk to sort of tie down some of these small gastropods and it will crawl inside the shell to feed. We have a set, this little white one in the bottom, of caterpillars that are specialists on fulgurids, which are plant hoppers, so the little hemiptera that feed with a piercing sucking mouth part. And then within our blues and our gossamer butterflies, we have an entire set of caterpillars that are either predatory or sort of parasitic in ant nests. What you're looking at is a Liphria caterpillar. So you're looking sort of at somebody holding the underside of it to show you all of its little feet. And then on the bottom image, what you're seeing is the caterpillar inside the ant nest actually feeding on the brood. So we have this enormous number of species that are soil feeders, moss feeders, root feeders, some of which are feeding on animal waste, and we have ones that are predatory or parasitic as well. And so all of these are things that I think most of you might not have been familiar with. One of the things that I didn't mention when I was showing that phylogeny is that given the timing of evolution of most insect groups, including Lepidoptera, what we know to be the case is that it occurred before the breakup of Pangea. So if I present a group that occurs in Australia, really what we have seen is a similar radiation of all of those groups across all continents prior to the breakup. So we can find pretty much all of these things within the continental United States. There's very few families that we wouldn't also encounter here within the US. All right. Now, when we get to plant feeding, some of the first things that we would have had that would have been plant feeders wouldn't necessarily have been like that large Cecropia moth feeding externally. It took a considerable amount of time before we had caterpillars that were feeding outside, exposed on the leaf surface. So these are plume moths. These would have been some of the first plant feeders, but what we see for this particular group is bud feeding. So they're feeding inside some of the tiny buds of the flowers, which are often much more nutritious than the leaves themselves. So we have a six-plumed moth here, one of the Montana species that's um, a feeder on, this is honeysuckle here. And what's interesting to me about these in the adult stage is that they have a very atypical wing shape for a moth. What we see is this sort of feathery plume. In this case, there's a 20-plumed moth that's in this image, and we think this is where we start to see some of this counter adaptation against being consumed by bats. So here we can see in the inset image is from a COLAB study in 2016. They were looking at the sort of echo that comes off the wing. So what's the shadow echo that comes back to the bat? And in the plume moth, what we see is that sort of reverberation doesn't penetrate as far. It's much more difficult for the bat to find these because they have those plumed wings as adults. But the caterpillars are super tiny. In the bottom, what we're looking at is one that feeds on the leaves of cacao. The center image is what the pupal chrysalis looks like. And I've just put an image of the chocolate bean fruit in here if you're not familiar with what that looks like. So the adults are pollinators of that particular plant. So the caterpillar is going to be feeding on the leaves and barely making a dent in the overall productivity. And the moths can be critical pollinators for this particular plant. But this is really the first type of plant feeding that we would have seen in the groups that are associated with our flowering plants. All right, now, for the most part, though, if I'm teaching a class on pollination, one of the things that I tell my students right away is that while we tend to think of butterflies as pollinators, really, that's really not what I'd like my students to come away with when they take an entomology course. And one of them recently made a meme. I don't know if you've seen the GIF that's going around of a guy holding his girlfriend's hand but looking over his shoulder at another woman. They put Lepidoptera up there and looking over the shoulder at other pollinators. So mostly what we see here is these tiny little moths that would be feeding internally on the fruits. As caterpillars, you can see those in the bottom image here on lychee. We have some on sumac and gooseberry as well. And the adults are these super tiny moths that are acting as fruit pollinators. So this is where we really start to see radiations on flowering plants. They tend to be single species feeders and really important as pollinators as moths. So we really think, particularly in tropical environments, moths are pollinating maybe 15 to 20%. Butterflies are a super tiny amount, maybe 2 to 4%. The rest are things like beetles and our common bees and wasps. Right now, as we get into things that are really feeding on the leaf, we're still not necessarily seeing things that are feeding externally. What you're looking at here is Caloptilia paradoxicum. This is one of the ones that we've nicknamed leaf folder head shaker because we didn't have a species name for it until recently. They fold the leaf over, hide inside, feed on the nutritive tissue. You can see the waste that they're leaving behind there. They even have a bird defense. So I pried the leaf open here, and I don't know if you caught that, but when it thinks it's about to be predated upon by a bird, it wiggles its little head back and forth to try to be threatening. It's really funny to watch something that tiny try to scare off a bird or a large person, a collector, an entomology collector. But I thought it was just some of the most amazing behavior when I first saw it. Now, what I'd like you to note about the leaf tissue here is that it is really sort of a foamy, a little nutrient dense. And so what we've discovered, and if you're familiar with galling wasps, is that we do have a very large number of caterpillars that can make gall tissue, very similar to some of these cyanipid gall wasps. So if you're out and about looking at oak, you may have seen some of these gall wasps. We know the caterpillars do this too. And in case of Caloptilia, what we have is a method of taking the microRNA from its saliva, so some of the RNA, the small nuclear RNA and mitochondrial RNA, and injecting it into the plant and sort of a subterfuge, making the plant make either reproductive fruiting tissue or more nutritive leaf tissue. So DPH here stands for days post-hatch, and you can start to see the larvae there in the center about day 9. It's kind of large enough to see. But what it's doing is using its own sort of genetic material to subvert the plant cellular processes to make this extra nutritive tissue. So Caloptilia is one of those that does that, and that's why it looked so white and dense on the inside when I opened up that leaf. Now, what we're finding is there's an enormous number of these microRNA that they're using to subvert the plant growth processes. So we have things that will make stem galls like our wasps do, or fruit galls, flower buds. So what you're looking at in the center right image is one that mimics the fruit of the vine on which it feeds. But we also see that they can force the plant to make mechanical tissue. So the little operculum, the door opening in the top there for the neospecia, is one of those things. So it makes a nice little hole, a fruit that sort of mimics the fruit, feeds inside of that, and then when it's ready to pupate, just pops open that little hatch and comes out and forms the chrysalis. What you're looking at in the bottom is Brazilian sumac and some of the galls that this particular caterpillar forms on that plant as well. And then we have the rose bay willow gall. Is also a caterpillar and not a wasp in this case. So we're discovering that more recently, as recent as 2023, that it's not just wasps that do this, it's caterpillars too. All right, but then what we have within that same group that's gall forming is one that I refer to as bark scribblers. So again, this might be an Australian version, but we have these here in the United States. You can just sort of follow the trail where the caterpillar's feeding right under the bark of the tree. And down there in the bottom, there's a circle. If you fold that open, you'll find the caterpillar inside. And you can see in this particular case, the mandibles on this ceciliosid are extremely large to chew through that bark. All right, now, I don't want to underestimate here the damage that some of our crop pests can do. So now we're moving into, in this sort of evolutionary stage, things that are feeding externally. So if you've tried to grow anything, whether it's on your porch in your deck, maybe in Arlington, I've tried to do it on my balcony in my small apartment. If you can avoid the squirrels and you can get anything in the Brassicaceae, broccoli, cabbage, spinach, anything to grow, you're probably going to find the diamondback moth and the cabbage worm moth. And these are going to be some of the first in the plutellid that would be some of our external feeders. But notice that they're tiny, they're green, they're very cryptic, and thus hard for the birds to find and consume. So they can do quite a lot of damage. But this brings us then to some of the more familiar types of caterpillars. So while we do have this enormous range of things that are feeding in these novel environments that we might not have known about, what I also want to talk about is that I tend to have this misconception that our caterpillars and our butterflies are these warm season types of things. So I want to dispel that. They do occur in all of our habitats worldwide, globally. So this is the Arctic Apollo or Siberian Apollo, Parnassus. And what we see in some cases, these particular groups have some of the longest lifespans. So in addition to thinking of caterpillars as being these sort of warm season things, we also tend to think of them as having very short lifespans. So when I worked in a butterfly house and the kids would come in, I would say, oh, they only live about 2 weeks, because what would happen is the children would bring me the dead butterflies and ask me to sort of resurrect them. So I had to tell them they have a very short lifespan. But in the case of some of our woolly bears and our Apollos, what we see is a lifespan that may be as long as 7 years. So this is the Arctic woolly bear. And what we see in this case is that it takes about 7 years in the larval stage. So you would have coming out in the summertime, feeding for a little bit, using those sort of antifreeze proteins in the body to keep it from freezing, coming back out again in the next summer, and taking 7 years to get all the way to that adult phase. The upper image shows you what the cocoon looks like for this particular species. Now, we do have woolly bears here within Virginia. Dave Wagner writes in one of his books about this particular family, the Arctiid, about this particular woolly worm festival that's held in Banner Elk, North Carolina every year. They claim to be able to predict the weather with about 50% accuracy. So, flip of a coin based on the coloration here of the woolly bears. What I'd like you to know about these local species here is that this sort of wandering behavior that you're seeing, and I've been seeing them out and about on my walks this week, it happens both in the winter, or rather the fall, rather, and in the spring. So they do two sort of migratory walks. One that we're seeing right now is just to wander away from the host plant and find a place to overwinter in the caterpillar stage. It may take them about 2 years in some cases to finish development. The spring is more in search of that host plant. If you take these into the lab, we thought perhaps this was a circuitous kind of wandering route, but in the lab, if you take away all the barriers and sticks and rocks and things that might be in the way, they will walk a very straight line. They may do that for as long as 48 hours before they sort of nestle in and stay still. So the adults here are some of the more common moths that you would see maybe at your porch lights in the evening. All right, so they are in some of our Arctic environments. They can have long development times. They're feeding on more things than we think. And I also want to mention, because we are talking about a marsh group of folks here, that we do in fact have aquatic and semi-aquatic larvae. So we don't tend to think about Lepidoptera as something that would be in the water like our caddisflies or mayflies, but they are. This is a white-tailed diver moth in this case, but we have some that are fully aquatic through almost all of their life stages. So we have a water lily leafcutter that does occur here, Virginia, and we have the cataclysta here as well. These have these life cycles that look a lot like our caddisflies. The bottom image shows some of the sort of feathery adaptations here for getting oxygen underwater. And then they come to the surface, usually extrude to form the chrysalis above water, and the adult is terrestrial and usually either in some cases non-feeding or nectar feeding or pollen feeding as an adult. So we have them in aquatic environments, we have them terrestrial, obviously, we have them underground as root feeders, we have them in both cold and warm environments. Now, this is showing that in some cases, we think that these aquatic habitats were able to evolve because of the sort of peristaltic movement that these caterpillars have. Actually, is fairly well adapted for swimming. So they sort of thrash their bodies both to get away from birds, but this also helps them to swim if they happen to accidentally fall into a puddle. So some of our species that even aren't aquatic can still make their way out of a pond or a stream if they actually fall in. So we do see some aquatic behaviors here as well. All right, now what I want to get into is again some of the misconceptions about why it is or that caterpillars look the way that they do. And what we're going to see is kind of a recent decision that most of the morphology, these colors and things that we see, are the outcome of two very different conflicting selection pressures. One of those are the plants that I've been discussing. So the plant toxins themselves we'll come back to in a moment. The other is all of their predators. So we're talking about predominantly here in the US, some of our passerine birds. Now, given the really high densities of a lot of these, what we can see is that caterpillars, when we have an outbreak, can increase bird density as much as 80 times. A lot of this comes from studies where we have tried to exclude birds from habitats to look at caterpillar increases, or conversely, we've tried to remove all the caterpillars, which isn't something that I would suggest anybody doing some from these older studies. So they are really supporting some of our higher trophic levels in our habitats. So when we do these exclusion studies, if we put a net even over a field, an orchard, some of our gardens and roadside habitats, or climbing up into a tree canopy, we see that when they're removed, they can reduce the radial growth of trees by about 47%. And I've set this inset image here of this sort of nutrient cycle, this carbon cycle in this case, because what we have been finding is that our climate models where we're trying to track all of the carbon, have had really what we wondered about was this sort of missing piece and what we called missing carbon. And what we're finding is that if we don't account for how much the caterpillars are eating and their role here in these forests, we're not doing a good job of estimating how much carbon our trees are actually able to store. So we can see that when we do these exclusions, whether it's excluding birds in some cases with netting, whether it's doing an ant exclusion because they do tend to have a pretty big impact on our caterpillars, we do find that it is an enormous amount that these organisms are able to reduce plant growth. So as we get into these, what we see is quite a number of organisms that are dependent upon caterpillars. So we have the Calisoma beetle here in Virginia. We have ants, a lot of our reptiles as well. We have our sphecids, so our potter wasps, some of our jumping spiders as well. This is a weaver ant nest here. But we do see that those are not the weaver ants, but ants in general here in Virginia are also predators of caterpillars. So they're supporting a large number of organisms and also are needing to sort of fight back against all of these. So as we start to look at why caterpillars have the colors and the morphology that they do, we see them as sort of the metaphor devil in deep blue sea. In this case, it's the devil in the deep green sea, the sea of leaves. So we have this need to overcome all of the sort of counter defenses of the plant, as well as to hide from a lot of these predators. So in the bottom, what you're looking at is a photograph of what the caterpillar mandibles look like if they're fed on artificial diet, which has all of the sort of toughness of the plant removed, versus something that's feeding on corn. So the mandibles do wear down, and they are replenished then as the caterpillar goes through each of these molts. So a plant may defend itself with a variety of toxins, resins. That's the classic example here is the poor little monarch caterpillar in the bottom left here getting stuck in that milkweed resin. So we think now that having to deal evolutionarily with both the plants and the predators is what gives rise to this incredible morphology and this morphological variation that we see in our caterpillars. So what I want to do is talk about how some of each of those different selection pressures has created some of what we see. All right, so we typically, this is the typical thing that might be in our head if we're thinking about sort of different instars that a caterpillar goes through. Again, it's the classic monarch. But what I want to convey here in terms of dispelling some of our misconceptions is that this particular looking exactly the same in each instar isn't really the norm for most caterpillars and most moths. Now, the extreme changes that they go through that I mentioned in the very beginning, though, do mean that what they're facing in terms of pressures to survive are very different for each of these instars. So, in the first instar, it might just be getting that first bite. And what we see in some cases is then the ability to eat the eggshell that you hatched out of as maybe a first meal. Instead, for a lot of other groups, then, what we see isn't necessarily that looking identical, but really big changes indicating different types of pressures and needs to survive as they get larger. So this is Acronicter funeralis, the funerary dagger moth. It goes through some of the more extreme changes. It might start out as a tiny, we call it a curly sue, a tiny little white almost see-through caterpillar in the first instar. It's going to be a bird poop as it gets a little larger. And the final instar I tend to see uh, potentially as a millipede mimic. So there's a millipede in the bottom left there. I often see people mistaking this particular caterpillar for a millipede, given that sort of mimicry there. And it's in this case, it's mimicking the toxic secretions of this milkweed to avoid being consumed by birds. So, one of the first things that you might do as a caterpillar, if you're faced with having to overcome some plant defenses, in this case, we're talking about some of the carnivorous, maybe the sundews that might be in some of our marsh habitats, is to simply be very, very careful. So, in the case of some of our sundew feeders, what we see is that they just move very slowly and crawl around some of these little trichomes with their exudates that are going to sort of catch these caterpillars, and they will reach up to the top of these and sort of chew down and bite off the things that the plant has to catch them. So feeding very carefully, eating your eggshell might be some of the pressures that we see, solutions to the pressures that we see in those earlier instars. The other thing that you might do is to try to circumvent some of those plant toxins. So for our milkweed, again, butterflies, our caterpillars are the classic example of trenching behavior. So you can see that this tiny little first instar simply ate a circle, and now all of the exudate is coming out beneath that, and it can feed on the tissue above and have exposure to much, much less resin. So we see the arrows here in all of these little images indicate where the caterpillar has first tried to chew through and block some of those toxins so that it's not exposed. Now, what we see sort of in the inset graph for the Thoroa here that's feeding on spurge is that it gets better and better at this at each instar. So the amount of resin is much, much reduced as the caterpillar gets larger. Now, what it might also do is feed on some of the early spring leaves. This is like baby lettuce for caterpillars. It doesn't have a lot of toxins. The plant hasn't the cellular mechanisms yet to make a lot of those horrible compounds. And so it can feed early in the spring as one way of avoiding that. Now, the other thing that we'll see as they get larger is they might actually begin to demonstrate some of these anti-bird defenses. So what you're looking at in the inset image is a photo I took of a skipper that is the caterpillar is cutting that leaf petiole to drop that leaf off the tree. So the evidence of its feeding in that damaged leaf is no longer on that tree. And this can sometimes lead to a large amount of green leaf loss in the middle of the summer when these outbreaks are larger. So one of the other ways that we see caterpillars dealing with some of these plant toxins is simply to have the enzymes like we do for detoxifying. So in the top image, you're looking at one of the Spodoptera, which is a common pest in a lot of things like sweet potato fields or soybean fields. The cytochrome P450 oxidases are the same types of compounds that we use to metabolize medicines that we take. The bottom image is showing you a silk moth where the gut is dissected, showing you how large that gut is for that particular caterpillar. So having that large gut is effectively similar to a ruminant, maybe like a cow, where you have a large digestion time that's letting you break down all of those plant toxins. So the inset graphs there in the bottom are showing from the starting mulberry leaves to all of those much smaller metabolites that are much less toxic as it goes through the gut of that silkworm. And what we're also finding here, particularly for silkworms, is that they also have some of the same things we do in our gut to help them detoxify, and that's gut bacteria. And in some cases, that can be developed into a probiotic to improve the growth of the caterpillar. And again, I don't want to underscore how really important caterpillars can be monetarily. If you've not ever seen the silk industry in China, this is what this looks like on an industrial scale. So having an ability to increase production or make the moths healthier in the caterpillar stage is really important. And I want to point out here, if you're not aware, silk moths are really considered to be our only truly domesticated insect. We think of our honeybees really as semi-domesticated. They're still free, sort of in their adult stage, flying about. But for the silkworms here, the females are flightless, and they are, at this point, a truly domesticated insect. Now, one of the other things that you might do if you can't detoxify this particular plant, if you can't trench it, is simply wait for the dead leaf to fall to the ground and consume it after the plant simply is no longer producing any toxic chemicals. My students here love the name of this. I'm sort of the post Harry Potter or before Harry Potter generation. So the Hermione here, the litter moths. These are sweet that are going to be feeding. So I would really encourage you right now not to necessarily rake or mulch your leaves if you can leave them laying out for a while for this particular set. So they're playing an important role in decomposition as well as reducing tree growth and serving as prey for some of our higher trophic levels. And this ability to both serve as a decomposer, a root feeder, and produce a large amount of waste has a pretty big impact then on plant growth as well. So I tell people that my brain is full of a lot of tedious information, some of which is the shapes of different caterpillar poop. I apologize if you're eating dinner right now. But just to show you, this is from some studies that I refer to as laundry basket studies. So we buy out all the laundry baskets in a Target or a Walmart. We set them out in the forest. We let all the leaves and plant material fall to the bottom, and we sort it out and we figure out what's plant material and what's caterpillar poop. You can weigh it You can dry ash weighted in a machine that looks at the amount of carbon and nitrogen, and you can figure out how much these insects are assimilating. So a caterpillar might only assimilate about 16% of the nitrogen, the protein in that leaf that it needs for growth. All the rest of it is then in this highly mobile form of frass. And when we have an insect outbreak, normally, when we're talking about a plant towards the end of autumn, reabsorbing all of that nitrogen through that senescence process, it usually reuptakes about 75%. But when we have a caterpillar outbreak, we're seeing only about 22% of that reabsorbed and another 22% returned to the forest floor as this caterpillar waste and this highly available form of nitrogen. In addition, we'll see an increase in that early leaf drop that I mentioned from leaf clipping. The insects themselves, in terms of their waste, their exoskeletons are leaving about 8% nitrogen. And the leaf that is upsized is adding some more to the soil. So they're important both for carbon cycling and for our nitrogen balance as well in our ecosystems through this consumption and then release of plant nitrogen. All right, so you can be a good decomposer and consume leaves after they've fallen. You can detoxify if you have the right gut compounds or bacteria. But what we also see is the ability to sequester. And by that, I mean to take up that plant toxin and to put it into your own body and to be toxic to things that consume you. So Glenda was asking at the beginning, and it's not really caterpillar season. Actually, you can probably find some of the slug moss, the lima-coated, this time of year. I'll show you some photos in a minute. These in the top here are our stinging rose caterpillar. In the bottom, we have our spun glass slug, some of our more interesting and brightly colored ones, really indicating their ability to take up these toxins. Now, both the lima-coated caterpillars and the megalopygid, here, ones that I think look a little bit more like mice than caterpillars, are able to take up these plant toxins. And in the case of our flannel moss, the Megalopygia percularis is one that occurs here in Virginia, as does the black flannel wave, which is the one in the bottom. I've seen an outbreak of it recently at Fort Valley. So if you go hike Manica Peak, for example, or Signal Knob, you'll see these this time of year. This is actually taking up the tannins in the oak leaf, and it will take them up into these hairs. They're not really toxic in those early instars, that's the upper image, but as they grow and sequester that tannin, it becomes as powerful as a dioxin type poisoning. So if you're familiar with the 2004, I believe, the ex-president of the Ukraine, Viktor Yushchenko, was poisoned with dioxin. This is that toxic. And this is a SEM close-up of what the hairs on these caterpillars look like. Some of them have these backwards facing barbs that are like fish hooks. And when you break them open, they're hollow so they can exude that particular toxin. So if you brush up against these, they can cause some pretty severe skin burns. So be careful of the later instars of these. So this definitely is a way, and a highly effective way, to defend yourself against predators. In fact, we have at least one example of a caterpillar here where the bird is now the caterpillar mimic and not the converse. This is the Cineros mourner. It's a Brazilian bird that mimics one of these flannel moth caterpillars. And then it's preventing its own consumption by birds of prey. So this is sort of a turnabout is fair play in this particular case. So this is a photo from Sam Jaffe of the Caterpillar Lab. If you've not seen his work, I would definitely suggest that you go online and look up the Caterpillar Lab, just showing you that this particular group, because they're feeding slowly late in the season and uptaking these chemical compounds, you could probably find them this time of year if you're looking at autumn leaves. Now, one of the additional ways that you can hide is to simply be camouflaged. And the interesting thing that we're learning about this is that depending on the time of year that you feed, you can still mimic different parts of the plant. This is Nemoria. This is an inchworm or a geometrid caterpillar. And what it does when it's feeding in the spring on oak catkins is look like the catkin itself. And when it's feeding on the green leaves a little bit later in the summer, it looks like a twig mimic. And you can actually induce this in the lab by feeding it the particular type of chemical mixture. So you can feed it more of a sugary gallotannin along with some protein, and it'll turn into the oak catkin mimic, or you can feed it more of the condensed tannins and it will turn into the twig mimic. Now, both look the same as an adult, but this plant-derived compounds are giving it this ability to change its morphology. Now, in some cases, it's not even the consumption, a different consumption, different diet that changes your color. It's that you perceive the environment as a different color. So this is the peppered moth. It's our classic example that we often use to teach students about evolution in the adult form, but it's turning out to be so much fun to experiment with in the caterpillar form as well. All you have to do is put dowels in the container with the willow leaf. Depending on the number of each color that you put in there, you can induce a change from maybe the whiter or the browner form all the way to the greener form. Now, the most fascinating thing about this recently is there was just a study that came out that showed that it wasn't actually visual perception through those tiny stomata that I mentioned that triggers this change. What you're looking at in the bottom image is on the left is a little circle that shows you the eyes, and on the right, it shows you where the researchers painted over the eyes of the caterpillar, the stomata. And so, what we believe is going on here is they actually have along their cuticle other extra optical sensory organs where they can sense some of the external environment. So even if you blindfold them, you still wind up with that same color matching in the environment. Now, in the event that you can't color match through some type of visual organ, extra optical or otherwise, one of the things that we see in the geometries, the inchworms as well, is decorating yourself. These are the fashionistas. I think this is the walking on the runway here with your particular flowers glued to your back. Synchlora tends to be one of my favorite caterpillars to find. I would definitely suggest if you have a garden in your backyard, looking at some of your sunflowers and false sunflowers or your New York asters. This is a good time of year for those, and seeing if you can find some of these decorator caterpillars. Now, in the event that you can't decorate yourself, we do see quite a bit of shelter building behavior in caterpillars as well. So you may be familiar with the silver spotted skipper. The bottom inset image is just showing you that as it makes these tent shelters and it gets bigger in each instar, the tent sort of gets correspondingly bigger for each of those. There's some argument to be made here that the face of this, the head capsule of this caterpillar is a snake or at least an eye spot mimic with the bright orange spots near the mandibles. But what I'd rather mention here is that there's quite a number of caterpillars that are shelter builders in other ways. They're not just folding the leaf, they're taking a lot of plant material. So we have our evergreen bagworm in the center occurs all across the United States. The female actually spends all of her lifespan inside that case. She stays the caterpillar stage, the adult stage as well, and simply averts her mating organs for the male from the case itself, and she'll lay the eggs inside the case for the next generation. We have the pagoda moth in the top. Yumeta is one of my favorite. It builds these little log cabins of perfectly stacked twigs. And then we are uncovering a variety of new things that still have yet to be named. In the center of the bottom is one that has used a crane's bill, the sort of winged seeds to glue on itself and carry around. So it looks a little bit like sort of a feathered seed in this particular case. So be on the lookout. You can definitely find the evergreen bagworms this time of year as the leaves start to change. Now, in the event you can't decorate with the plant itself or some other plant material, you can keep your old exoskeleton. So we have some that are nicknamed caterpillars because they keep their old head capsules. They stack them up, and these turn out to be pretty effective at deterring some of the stink bugs. So we've had some recent lab studies that show that as they're wandering around, they'll point these little head capsules that are stacked up here at the stink bug and help deter that particular predatory insect from feeding on them. Now, in this particular case, what we also have is a defense where group size also matters. So if you can't deter your predators or hide from your predators, you might seek safety in groups. So we're looking at the group size. Can this particular caterpillar can be as much as 50 individuals. As we start to see, as they're in these sort of smaller groups, though, they rely more heavily on those stacked head capsules. So one of the things that we also tend to see is in addition to being cryptic or mimicking something distasteful, is that the caterpillar might actually also be distasteful. So this is Mae Berenbaum's classic study, where what she looked at was the fact that the uric acid, what's in the sort of urine and waste, is actually more concentrated in the center portion of the caterpillar more. So if a bird does try to consume this, it not only tastes or looks like bird poop, it tastes like that as well. Now, for the swallowtails, this is a giant swallowtail here in this image. What we see is that moving from a sort of maybe spiny, tiny instar to a poop mimic in the middle instar is fairly common, and the trajectory then is to become a snake mimic with big eye spots in the last instar. So the pipevine swallowtail is one of my favorite ones for my students to search out this time of year. So this is showing you in the sort of additional papilio species, swallowtail species here, showing you those early instars being that poop mimic and the later instars having these eye spots. In the bottom, what we have a variety of swallowtails and other moth caterpillars, so things in the sphingid, for example, that are more tropical in origin, all of them being snake mimics. Now, in some cases, what we also see, and this is particularly true of the swallowtails, are additional organs that will evert from the head capsule and secrete fairly toxic chemicals. These are called osmeterium here. So we're looking at all of these across a range of our swallowtails here. They avert this by sort of contracting and increasing their body pressure and pushing out. It's almost like inflating a balloon. These smell really nauseous, it's a very acrid vinegar type smell. They do deter birds, and there's been some additional studies recently to show that they're less effective, though, at some of our stink bugs. So what we might also see then, if you can't sequester the compound or cover yourself in the plant or be toxic, is that you look like the predator itself. Now, the common name of this caterpillar in the upper left is the lobster moth, but in this particular case, if you look at the image of it from the underside on the right, it really is more of a stink bug mimic, so it's mimicking one of its own predators. And we have things that are snake mimics that aren't just this sort of face of the snake that actually do mimic the snake patterns as well. And we're discovering a variety of ones, this is Naomi Pierce's work here, that are also ant mimics. So if you can't hide from them, look like them is the other approach. Now, I did want to show you how effective this group defense is. So what you're looking at in the upper inset graph is the survival curve, and that upper curve is showing the much greater survival for these groups that feed together. So feeding together is not only a mechanism of sort of dispersing the sort of predator pressure among your siblings, but it also does help you break into that leaf tissue of all of you are sort of secreting your saliva and chewing on that leaf together. So for Semarista, this is Lochmaeus monteo, the variable oak leaf caterpillar. For some of our rosy maple moss as well, that's the upper image, we'll see that group feeding is common and that they'll stay in those groups throughout some of their instars. Now, this group feeding then can also be a cryptic defense. So this is one of the morpho, the blue iridescent butterflies from the tropics, mimicking some of these chenille type or mimosa type flowers on its plant as a group. So the group has to stay together here to have that effective defense. Now, what we see then when we get to these brighter, odder sort of displays is a combination. These caterpillars are using everything they can to defend themselves against the plant and the predators. So in this particular group, Datana and Semarista, these are some of our nododonids, our sort of larger headed caterpillars. We see group feeding behavior. We might also see at the same time that they regurgitate and spit up some of the plant toxins. That's the image in the upper center. They might also secrete some of their saliva. This is red Semarista saliva from Gisard's work here, showing that it sort of wipes its mouth parts on the plant petiole to secrete that salivary enzyme into the plant and break down those plant toxins. Then we see some leaf cutting behavior to hide its damage from birds. But what you're also looking at is this very large gland that's underneath the head capsule that can secrete a very acid compound to break through the leaf petiole. And then in the bottom image, what I've included is an image, a close-up of the caterpillar silk glands. These are on the mouth, not on the sort of abdomen like we see in spiders. And the upper image, the LS, is that labial silk gland, showing you how long those are and how large they are. So silk is a pretty important evolutionary origin here in caterpillars. We actually see this in a variety of other organisms. So we're going to see this as well in some of our marine organisms. It evolved fairly early on prior to when butterflies and moths emerged. So we often see group feeding. Maybe you see some tent caterpillars on cherry. This is the time of year that we might be seeing fall webworm. And the upper inset image just shows you again some of that group defense, that startle mechanism being fairly effective as a predator defense, so that if you're in that silk and you can make this, you sort of startle your predators away. Now, silk itself can be used a little bit more simply. This is one of our basket weavers. This is Lacosoma. It's actually hiding inside the leaf that it's consumed. It's taken its silk and it's wrapped it up with its own frass to make what looks like a little basket to hide in, and you can open that up and find the caterpillar inside. So if you're looking at dead leaves this time of year, this is another silk using caterpillar here that's using this sort of silk as camouflage. Now, again, this is the diagram here showing you the evolution of silk. So it's there in our very primitive insects like silverfish and bristletails. We see it in grasshoppers, but we're talking about it all the way down here at the very bottom of our chart in our caddisflies and our butterflies. And so we see in this case that we have a variety of uses for silk. We might be building a cocoon. That's how we typically think of it in our butterflies and moths. But in the case of our tiny little bucolatrix, in the upper right image, you see the tiny caterpillar feeding underneath a very sort of saran wrap layer of silk, and then it builds a cocoon and almost a little fence around that to prevent it from being consumed. We'll also see that because it evolved around 140 million years ago in Lepidoptera, that it's used for dispersal in a lot of organisms. And this may include inter-island dispersal in some of our archipelagos. It's used for communication. So you're looking at a processionary strand here, processionary caterpillars following that silk and the chemicals. But the fun one for me when I'm talking and teaching to my undergraduates is Stichomeris. This is a common one in Virginia. It will use the silk to fold the leaf over and feed inside, but then at the end of the season, it crawls up, it cuts the leaf petiole, and it sits right in the leaf where the aerodynamics is such that it's almost like it's made its own paper airplane, so that rather than just dropping to the ground, it can actually somewhat steer the leaf away from the parent tree and away from the predators. So it really makes its own sort of magic flying carpet here in this particular caterpillar with the use of silk. Now, I want to end with some upbeat things, again, emphasizing some of the economic value here of caterpillars, because this is one of the more derived, more recently evolved silks rather than the primitive ones that we see in some of our other insects. It has a canonical structure, meaning that it's more tightly wound than we typically see. And it is turning out to have a lot of properties that make it really great in medicinal research. So it doesn't tend to have the same level of rejection that we get for a lot of tissue grafts. So it's compatible with your blood, has immune compatibility with vascular cells, so reconstructing some of our veins. We're using it for wound healing, for sutures, for bone tissue engineering, and also looking at the use of it for drug discovery. And I'll end, I guess, with one of the more prescient or important things of our time, and that's the use of caterpillars in the preparation of vaccines. My mom has an egg allergy, so vaccines were always a problem for her. If you've ever seen the preparation of vaccines in eggs, in large scale, this is what that looks like. So you're injecting it, you're letting it incubate inside that egg, and then harvesting the vaccine as it's replicated. And what we're finding, and this is studies that date around to 2020, so right before the pandemic, is that those egg-based processes, which we have researched really well, take about 6 months. Now, in development are some mammalian cell-based processes, and these include, for example, using dog cells grown in a lab, so kidney cells, to grow the viruses and make the vaccines. But what we're finding is that caterpillars, like some of our crop pests, so Trichoplusia, our cabbage worm, and Spodoptera, our sweet potato worm, can also have bacilloviruses that can be used to replicate our vaccines that we make. And that if we are using them, the process then can be sped up to about a month. So this is some of the newest and more interesting ways that we're using them in an economic setting. And I'll stop there. I know I've been talking for a while here, and I'll take anyone's questions. And I apologize for going over here on my time.
Not at all. That was amazing. Thank you so much.
Mhm.
We do have a couple questions in the chat, and I'll do those first. And then I'm sure there are others that may have a few questions. The first is, what should we do to support caterpillars? And a follow-up to that is, can you offer some tips for spotting caterpillars?
Yeah. So we'll start with this tips for spotting caterpillars. So what this because this again this I take students in the field all the time, and it's really difficult to find them, and they get disappointed if they don't find that giant caterpillar right away. So what I typically tell people is search like a bird. You're going to turn over the leaf and you're going to look in the bottom. And typically the more common species, so things like monarchs, may actually take a little bit longer. You may have to search more plants than you think. But I would say don't be afraid of walking out into the weeds. Don't be afraid of in areas where you can walking off trail and turning over and looking at the bottom of the leaves. So they're going to be usually hiding on the underside somewhere. Look for leaves that are wrapped up. Look for the damage holes as well. So birds often look and search for anywhere there's damage, which is why they've evolved those clipping behaviors. Um, but in terms of supporting them, I think, protecting habitat is about all we can do at this point. I would say for most of the butterflies, if you're planting your own gardens in your backyards, if you can. But keeping the leaves at the end of the season, I'm fighting my own HOA on that right now, probably is one of the things we can do to support a lot of these smaller moths. They're going to be cocooning in that dead leaf material this time of year. So that's a great question. Have patience. That's definitely what I would say. And I'm getting to the point where I need my bifocal. So bring a hand lens or magnifying glass with you to find some of the smaller ones.
We have a lot of compliments in the chat. If anybody else has any questions, please feel free to either put them there or just turn your microphone on and ask if you'd like, or you can use the hand raise button, whatever is easiest for you.
Yes.
I'm not seeing any right now, but oh, we've got one new message. Let's see. For my 6-year-old son. Aw.
How do they make their
How do they
silk?
make their silk? Yeah.
Okay, so they are making it in their mouth. So they have these large glands that go in the back of their mouth all the way down their body into effectively what is where their legs are. It takes up about the front half of their body. And it's a liquid form initially. It's multiple proteins. And then it starts to make that sort of very Spider-Man-like, if you've seen the movies, as it shoots out in liquid form, it starts to crystallize and become this sort of very elastic hair-like structure. So they're making that from saliva in their mouths. It's a great question.
Neat. Yeah, I didn't know that, so I'm glad that was asked.
I have one. As someone who's also trying to fight their HOA, I wondered if
Yeah.
you could elaborate on what you're trying to get them to let you do with the leaves, or if it's in a
Right.
common area, or what you're trying to do there.
Yeah, well, so they most HOAs have a crew that mows, and so they're going to be removing whatever is in the common areas, but leaving the leaf material intact in your flower beds or underneath the trees themselves. So a lot of neighborhoods are sort of forcing people to buy mulch or put the sort of real chunky pine mulch that's colored brown or reddish. And instead, if you're leaving the leaves and putting them underneath the tree itself, you're keeping the nutrients there. It percolates more water, and you're leaving all of the caterpillars that would be cocooning, or rather moths that were cocooning in that leaf material. So if you can just pack it in the flower beds. I know that people often don't want to put leaf material near their houses because they're afraid of encouraging things like termites, but I think that risk is fairly low relative to the number of species you'd be protecting. So leaving the leaves in your yard wherever you can is probably my biggest recommendation.
We have one more question. The caterpillars that we see building tents, are they problematic?
Say again, are they?
Are they problematic?
So you're gonna get conflicting comments on that one. So the ones that we see in the spring are usually our tent caterpillars, and they have a preference for cherry. And cherry is a DC spectacle, and so we tend to dislike them. My aunt asks me every year, how do I get rid of them? For the most part, they're not necessarily going to have a big impact on tree growth. So they may move off that tree and feed somewhere else as they get to the later instars. So unless you are in a nursery or you're trying to get a small tree to get started, then those particular species, the tent caterpillars, the Malacosoma, that are feeding on cherry, there's two species, there's eastern tent and there's a forest tent caterpillar, are usually going to disperse in later instars and not do too much damage. The Hyphantria cunea, which is the fall webworm, is the other one that you're seeing. It tends to be more scary than you would think, and that's because it likes the edge habitat. So as you're driving maybe down 66, you might tend to see them all on pecan or some of the other trees that are growing along the edge of the highway. And so we get this impression that they're in more abundance than they are. But the same thing is true for those. They may defoliate a whole branch. They may defoliate at the end of the season on that tree, but it's had a whole summer to kind of get all the nutrients that it needs. And so the damage they do, unless you're trying to get a small sapling to get started, really isn't something that would be problematic. And they tend to not really respond to sprays. So the things that we have to treat trees are systemic poisons that are going to hurt other things. Now, the one thing that is problematic are the spongy moths, formerly called gypsy moths. Those can do an enormous amount of damage, and we do have control programs for those. They don't tend to web, but they do tend to consume enough to cause some tree mortality.
There's
Yes,
a follow-up to that.
yeah.
Someone wrote, I used to see a lot of tent caterpillars, but not so many nowadays. Do you know what has happened to them, and what do they become as adults?
Yeah, the moths are sort of nondescript. So they're medium-sized moths as adults, night flying. You probably have seen them. I don't have an image here to show you. You probably have seen them around the porch lights at night if you're in a DC area. But I don't know if we are seeing the sort of general decline that we're seeing across all insects groups with those, or we're just maybe in a downward trend in the population growth for those. We are in what we consider to be an insect apocalypse, so a decline of about 60% in the populations of most organisms. And that's probably contributing to some of the decline that we see, like fireflies and other things.
Someone asked, claims are made that the spray controls for spongy moths also affect many other species. Is that true?
Okay, it is true. So they can be specific to the Lymantriid, which is the family in which the spongy moth sort of resides, has been appointed. But what we are looking at in some cases are also sort of a confetti-like mating hormone disruption. So it gets all over the wings of the males, and the males think the other males are females, and so they mate inappropriately. So we are looking at some that are more specific, but what we find is that because some of them previously have been more broad spectrum sprays, so I would say we initially started with DDT, is that what we're finding is that at least in treating that outbreak, you're saving enough of the trees that you are seeing higher diversity of the subsequent organisms. So we actually have to stop that outbreak in order for there to be enough leaf material for other things to consume. So Ray Mandarino has studied this. She was in Kyle Haynes's lab at UVA and has shown then in some cases, we get higher diversity when we actually do treat for those organisms. Now, what we do know is that all of the wasp parasitoids that we tried to release to control them initially, some of those have become established and have led to about a 60 to 70% decline in some of the native silk moths as a consequence. So there's some positives and negatives in that overall. Treating really is about, I would say, pretty necessary at this point just to save the forest themselves.
Yeah.
Anybody else?
Well, I'm not seeing any more right now, but thank you so much. This was a
Thanks.
fabulous presentation.
Thanks to
I
everyone.
know I learned a lot, and there's many compliments in the chat of people saying what an incredible presentation it was.
Thanks. I hope that everybody saw something new today that you didn't know. I hear there were some entomologists in the audience, so feel free to email me. And if you are interested in any of the background research on any of these or where the images came from and want to know more about the research, please do send me an email at work.
Thank you.
Thanks.